everyone, I am Dr. Sara Kashkouli from the Department of Anatomy, and welcome to our first embryology lecture of this academic year. So over the next 2 years, as you make your way first through phase 1 and then through phase 2, I will be taking you through the development of a human in a series I like to call How to Build a Human. Now, the entirety of this journey is going to take you right from the beginning of development, or embryogenesis as we call it, all the way through, the super intricate orchestration of each organ system in the human body. So as we go along in the next 2 years, with the start of each new system block that you encounter, I'm going to come in to lay the groundwork for you and to discuss how the development of that particular system takes place. Now, today's lecture in particular, in discussing embryogenesis, it's going to take you through those first 3 weeks of development, and we'll discuss what are the major things that are occurring during this time. So let's jump right into it. This is part 1 of How to Build a Human, looking at embryogenesis. So this is the general roadmap we'll be taking for today's lecture. So we're going to start right off the bat with a simple introduction into embryology. Why is embryology important? What does it mean and why do we study it? Whilst we're here in the introduction, we'll also discuss some of the new terminology that we're going to be using, not just during today's lecture, but in future embryology lectures as well. Of course, a lot of these terms will come in handy as you'll encounter them over and over again in your studies of gross anatomy as well. When we're done with the introduction, we'll look at the key events that are happening in the first 3 weeks of development. Now, the 3 key events in those first 3 weeks of development are fertilization, implantation, and gastrulation. Now, implantation is of course missing from this roadmap, but we'll briefly discuss it just kind of broadly to let you know how the process of implantation actually happens. But we'll spend more time talking about fertilization and of course the main event in the first 3 weeks of development, which is that process of gastrulation. And then once we understand gastrulation a little bit, when we've expanded enough on gastrulation, we can talk about those germ layers that we have, how those germ layers develop, and what they give rise to. So what the derivatives of those germ layers are. And when we've discussed all that, when we talk about how, we go from fertilized to implanted to then gastrulation having occurred, when we have the basic sort of template for creating, for a human to then go on to develop, we'll talk about embryonic folding as that developing embryo, starts to fold and actually take on a more human-looking like shape. And then finally, at this stage, we'll introduce the process of ossification. Ossification is the process of bone formation, and we'll come back to this when we look at the development of the musculoskeletal system in your second year or in phase 2 of your syllabus. But we'll do a bit of an introductory kind of laying the groundwork of what ossification means at this stage as well. Now again, I know a lot of these terms are entirely new to you, but I wouldn't worry too much about it because we will definitely define them as we go along. And of course, ultimately, when we talk about these 3, when we talk about the key events that are happening in those first 3 weeks of development, we'll also relate some of the clinical implications of what could possibly go wrong during those first 3 weeks of development. And if something goes wrong, how could that present in a fetus or in the baby afterwards? Now, to clarify the objectives of today's lecture, by the end of it, you should definitely be able to do these things. So you should be able to discuss the major events of the first 3 weeks of development. You should be able to describe each individual developmental process. So this includes things like cleavage, blastocyst formation, and gastrulation. Again, these are new terms to you, but we will define them as we go along. You should be able to describe what the germ layers are, what the significance of the germ layers are, and what the derivatives of each of the germ layers are. And derivatives, of course, just means sort of the things that those germ layers give rise to. Having done that, you should then be able to discuss in quite a fair amount of detail the importance of embryonic folding, why it has to happen, how it happens, and why it's important. And then finally, you should be able to differentiate between the types of ossification, which are intramembranous ossification and endochondral ossification. Okay, so let's start by introducing today's topic. So embryology, right? Or more specifically, embryogenesis. Now, what is embryology, you might ask yourself? So embryology basically tells the story of how life begins as really just one cell that goes on to get fertilized, and that fertilized structure or cell then goes on to evolve to become you and I, which are these super intricate and fully functioning human beings. And so to be able to achieve this really quite large transformation, that initial cell or that fertilized zygote, as it's known, has to proliferate, and it also has to differentiate. So the cells within it also have to differentiate into many, many, many new cells. And then these new cells are what go on to form those increasingly complex structures in a very precise, very organized, and very controlled manner, much like if you think about a very meticulously choreographed dance, wherein every single one of the steps is very, very carefully planned and then executed very flawlessly as well. So embryology tells the story of us humans and how we develop from a zygote that continuously proliferates and the cells within it differentiate until we start to change shape. So slowly we start to look a little bit different. So we change shape and structure a few times along the way until ultimately we transform into fully functioning human babies that are able to sustain life outside of the uterus that we develop in. And of course, what's important about embryology is that a good understanding of embryological development is going to help us lay the basis for understanding a lot of the major topics you're talking about during your next 2 years of medical studies. So it helps to lay the basis for gross anatomy, for physiology. It helps you understand pathology a little bit more because we talk about developmental anomalies as well. And of course, if any one of these moves along any point in this developmental process, so whether it's during cell proliferation or whether it's during cell differentiation, if any one of these things doesn't occur in the sort of precise time and manner that it's meant to occur in, that could lead to congenital anomalies, and we'll discuss that as well. Now, before we progress any further, I'd like to introduce some terms to you that some of you, of course, might be familiar with, and if you're not, now would be a good time to start to take note of some of these terms because you are going to repeatedly encounter them whilst you study embryology, and of course, definitely during today's lecture. So we start off with a zygote. A zygote is basically, in essence, the inception of life itself, right? So it's this newly formed cell the moment the fertilization of an oocyte or the female egg takes place by the male sperm. So that's the zygote formation. That zygote then kind of, as a result of some changes, which we'll discuss in the next couple of slides of the lecture, that zygote will eventually go on to become an embryo. So in the early stages of human development, we're in the state of being an embryo. And ultimately that embryo, again, as it develops further and further and further, will then become a fetus. So a fetus refers to what the embryo becomes next. So as what is known as the embryonic period of development kind of wraps up and concludes, the developing human is no longer referred to an embryo, but rather it's going to start to be referred to as a fetus. And by the time which we start referring to this developing human as a fetus, it's starting to be a little bit more recognizable as well. So it takes on a lot of the more sort of human features that we are familiar with. And you'll understand this, of course, as we go along. Next, we have gastrulation. Gastrulation is the formation of a blastula to a gastrula. Now, again, this is the broad definition of it. I know terms like blastula and gastrula are going to be new for you as well, but these terms will define as we actually go through today's lesson. So the gastrula is basically the more sort of intricate structure that gets formed. The blastula is a little bit more simple. The gastrula that is formed as through the process of gastrulation is what is the more intricate, more complex structure that is formed. And finally, it's as a result of this process of gastrulation that it gives rise to these 3 germ layers. And these 3 germ layers are super important because these are basically what give rise to exactly everything else in the human body. So we have the ectoderm, the mesoderm, and the endoderm. And we'll discuss all of this in detail as we go along today's lecture, of course, and then we kind of keep coming back to it in future embryology lectures as well. So ectoderm describes the outermost germ layer that's playing an important role in basically giving rise to or shaping a lot of the external structures of the human body. When we talk about mesoderm, it's essentially the middle germ layer that exists, and this is going to contribute to the development of things like the muscles in our body, the blood vessels, and by and large, it just plays quite an important role in the body's architectural framework. And lastly, of course, we have endoderm, which is the innermost germ layer, and that's going to contribute to a lot of the internal structures, like parts of the digestive system and the respiratory system. So these are the sort of key words that we keep discussing through today's lecture, and of course through future embryology lectures as well. Now, when we study embryology, again, so we said basically embryology is the study of the differentiation and the growth of an organism that it undergoes, through the course of its development from that single fertilized structure to being this highly complex, independent being, essentially like its parents, right? So when we look at the entirety of this process of development, in order to grasp it a little bit better, we can start to divide it into a few distinct blocks of time. And of course, each of these blocks of time bear their own significance. What we can do firstly, and this is sort of the more popular, more common way of dividing it, is to divide it into trimesters. So the first trimester, the second trimester, and the third trimester. And each of these trimesters, of course, has its own significant milestones. So the first trimester is basically characterized by the events that are going to start to lay down the groundwork for development, and it's also going to establish the basic bodily structures. All of that is happening in that first 3-month period, or in the first trimester. The second trimester is going to be characterized by a period of great progress, wherein that developing fetus is going to get more and more recognizable. A lot of the organ systems come in, a lot of the facial features come in, and this stage is marked by very rapid growth and increasing complexity. And the third 3-month period, or the third trimester, is when all of those organ systems that have already developed, they're going to basically continue to mature, and they're going to start to prepare to be able to sustain that baby's life outside of the uterus. And Of course, dividing it in this manner into trimesters is typically how obstetricians would commonly divide that 9-month gestation period or that 9-month period of pregnancy. Now, another way to look at it, and this is mainly how we look at it when we talk about embryogenesis or embryology as a whole, is to look at it in two main blocks of time. So we have the embryonic period of development and the fetal period of development. Now, the embryonic period of development is going to be the period that occurs between the moment of fertilization, so when that zygote is formed, all the way through to the end of the 8th week of development. So from day one or at the moment of fertilization all the way through to the end of the 8th week of development, that is known as the embryonic period. And this is largely the period we're going to be discussing today, because this is the period that includes the establishment of those 3 germ layers. Remember we spoke about what those germ layers were, the ectoderm, the mesoderm, and the endoderm germ layer. So that's happening during this embryonic period. And of course, it's those germ layers that essentially go on to give rise to the tissues and the organ systems in the body. It's also actually during this embryonic period of development that most malformations that lead to congenital issues actually occur. And like I said, we'll discuss those as we go along. So that's the embryonic period of development. So again, from the moment of fertilization or zygote formation all the way through to the end of the 8th week of development. Next, we have the fetal period of development. And the fetal period is basically considered as the period from the start of week 9 of development all the way through to the moment of birth. And it's within this fetal period of development that we have a lot of maturation of the tissues and organ systems within the body. So these are the 2 main chunks or 2 main periods that we talk about in the way of categorizing development. And so it's worth it to know, to basically note that at this stage, we talked about there being trimesters, and we also talked about there being this 2 chunks of time that we discuss. And this is because embryologists and clinicians work with slightly different timelines when assessing developmental phases. So for example, where an embryologist would use the point or the date or the moment of fertilization as their starting point, a clinician would instead use the date of that individual's last known menstrual period to determine the fetus's gestational age. And so that's essentially what introduces that 2-week discrepancies in an embryologist or a clinician's timeline. And so it means that if as embryologists we say that the embryonic period ends here, so it ends at the end of the 8th week of development, a clinician would instead report the end of the embryonic period as the end of the 10th week of development, because we have that 2-week discrepancy. Okay, so let's take a minute to orientate ourselves. So in gross anatomy, we describe the anatomical position as this one over here, right? So if you had to kind of verbally describe this anatomical position, you talk about it as being the standing position. So with the body upright, the feet sort of together, flat on the floor, slightly apart, with the body, of course, being upright, as we said, both arms on either side with the palms facing forward. The head is level and the eyes are looking forward. And in terms of, so that basically describes the anatomical position, right? In terms of directional terms that we use in gross anatomy in particular, we use anterior, posterior, superior, inferior, medial, and lateral. Now, anterior refers to anything that's ventral or at the front of the body. On the opposite, you have posterior, which is anything that's dorsal or to the back of the body. Superior, or another term for it, is cranial, describes anything that leads more towards the region of the head of the body. An inferior, or another term for it would be caudal, describes anything that leads toward the feet, or the inferior aspect of the body. Now, if you imagine a sort of central midline running down the body, anything toward the midline of the body is said to be medial, and anything sort of lateral or further away from the midline of the body is said to be lateral. So these are basically the directional terms we use in gross anatomy. Now, how this differs in terms of a developing fetus is that developing fetus, for the most part, while its terminology is quite similar, especially when kind of talking about directional terms, what's important to realize is that the fetus basically takes a sort of different kind of anatomical position as it's developing, right? So this is what a developing fetus kind of looks like while it's in utero and developing during pregnancy. So it's going to be curled inward with this back region, or the spinal region, flexed because the back is going to be curved. The head region of the fetus is going to be bent forward and slightly kind of tucked in towards its developing chest region. And of course, its little developing arms and little developing legs are also going to be drawn up toward its torso. So that's essentially what it looks like, and that's how we descriptively describe the position that the fetus takes as it's developing in utero. In terms of directional terms, essentially a lot of the same terminology is used. So you still have anterior, posterior, superior, inferior, medial, and lateral. Again, anterior, also known as ventral, refers to anything that's in the front of the body. Posterior or dorsal refers to anything that's in the back of the body. So dorsal is here, ventral would be anything here. Superior or cranial refers to anything that's toward the developing head region. Inferior or caudal is anything that's toward the tail region or the feet region of the developing embryo. And again, similar to in gross anatomy, medial and lateral are if you kind of imagine a sort of central midline running down that developing fetus. Anything that's closer toward the midline is going to be medial, and anything that's kind of further away from the midline is going to be lateral. Now, a lot of the things that you see on this slide, we won't spend too much time on because they're things that you come back to when you discuss the reproductive system. But the thing is, we cover the reproductive system in phase 2. So you're going to be studying it around about now in a year's time. So for the purpose of having to basically set the stage for today's lecture, I'm going to give you just a super brief overview to discuss kind of what's key, what's important that's happening here. And it's just so you understand the big picture of how fertilization even occurs. So this slide in particular is less about knowing the nitty-gritty details, but rather it's more about understanding the general principles of how we go from, single cells, so egg and sperm, to becoming a zygote. And so I'll spend the next few minutes basically, individually summarizing the journey of the egg, and then summarizing the journey of the sperm. So we'll talk about those separately before summarizing what happens when the egg and the sperm meet, and then how that leads to the formation of a zygote. So we'll start by taking a look at the journey of an oocyte, or a female egg. So if you look at this sort of illustration of the female reproductive tract, so here kind of depicting the uterus, here depicting the uterine tubes, and here depicting the ovaries, so one on either side, of course, another uterine tube here, and just the main uterus here. Of course, this is just a cartoon, so take it with a pinch of salt. But largely this is what it looks like. This gives you a good visual of what it looks like. So within the ovary itself, there are going to be many, many thousands of follicles, right? And each follicle, what that means is that it's basically a sort of hollow ball of cells that has an immature egg sitting kind of in the center of it all. As these follicles, as they start to mature, they're going to secrete hormones into the bloodstream to start to prepare the lining of the uterus in case a pregnancy occurs. So by the time you get to around day 7 of the menstrual cycle in women, the follicle within the ovary is going to stop growing, or rather, all the follicles within the ovary that were kind of growing stop growing, and they start to degenerate with the exception of one follicle. Now, that one dominant follicle is going to continue growing, and it's going to continue to nourish that developing egg within it. And so right before ovulation occurs, this egg is essentially going to detach itself from the inside of the follicle. And as that's happening, the follicle will release some chemicals. So these uterine tubes are not connected to, they're not stuck to the ovaries. So what happens is that that release of chemicals from that follicle is going to prompt these uterine tubes to essentially kind of move in closer toward the ovary and to kind of start to surround it, because eventually the follicle is going to swell. When it swells to a certain point, it will burst open. And when it bursts open, it is essentially is then able to kind of eject that developing egg from within it. Now, in response, the sort of end parts of the uterine tubes, these are known as fimbriae, so F-I-M-B-R-I-A-E. These fimbriae, which are these sort of tiny little projections at the ends of those uterine tubes, they're going to sweep across this general site of ovulation to pick up that egg. So they're kind of like these finger-like structures. They come forward and they sweep up that egg within it. Now these uterine tubes, they also have what is known as microscopic cilia, microscopic because they're small, and cilia, which are these tiny sort of hair-like structures that line the fimbriae surface to kind of create that sort of, because if you kind of imagine that egg moving, to kind of create that sort of movement, that brushing, sweeping movement to start to kind of transport the egg first in toward the uterine tube, and then, eventually when that egg is within the uterine tube, you're going to have muscular contractions that occur as well, and this will gently start to push this egg. If you kind of follow my pointer, it'll basically push the egg down through the uterine tube to start to try to reach the uterus. At the point, from the point of ovulation, the egg basically goes on to live for about 12 to 24 hours. So it has that little gap in time wherein it has to be fertilized by a sperm from the man for a pregnancy to take. So that's what's happening individually on its own in the female reproductive tract. Now, what's happening with the sperm? So if we look at the journey that the sperm goes on to take, we're essentially looking at the other end of the female reproductive tract. So coming in, if you follow my red pointer from this outside up through the vagina, the cervix, to get to the uterus. So at the point of sexual intercourse, somewhere around 300 million sperm are deposited into the vagina. And what this does is that it essentially basically starts to create a little competition from which only one sperm can emerge victorious. So if you follow my red pointer, so in the vagina itself, right off the bat, from these 300 million sperm that were deposited here, millions of sperm are going to die because the vagina has an acidic environment and the sperm just cannot survive in it. Now, whatever remains is going to swim upwards here through the cervix to start to get toward the body of the uterus. And what's happening by the time it gets all these however many millions of sperm that remain, when they get into the body of the uterus, these walls are going to have, these muscular uterine walls are also going to contract, and they'll help to kind of drive or push the sperm sort of upward so that it continues to swim up in this general direction. As the sperm are trying to do that, many more millions of sperm are now going to be killed in this environment by the white blood cells in the female's body, because of course the female's immunity is kicking in, right? It's noticing that there are these foreign entities within the body of the uterus, so it sends the white blood cells to kill these foreign entities, which are the sperm. From the ones that do survive, and they eventually get to about this point where my red pointer is at, they're basically now at a fork in the road, right? So we've got two uterine tubes, and this is essentially the fork in the road. So some of them can go to the left, and some of them can go through towards the right. But if you remember, only one of these uterine tubes is going to have an egg in it, right? So the half that, for example, will go down the wrong uterine tube, they're just never going to encounter an egg, right? And the other half are going to actually swim up the correct uterine tube. And so by the time you get to this moment where you have sperm within the uterine tube, you really only have a few thousand sperm that remain. And once you're left inside the uterine tube, remember you had the cilia, right? We had that microscopic cilia that we spoke about, and that microscopic cilia is kind of what's helping to push along or brush along that egg down the uterine tube toward the uterus. But then in order to successfully reach the egg, the sperm has to kind of swim up against that motion to be able to reach the egg. And of course, as you might expect, some of those sperms are going to get trapped within the cilia and ultimately die. And so what's also happening is during sort of this part of the journey where we're getting closer and closer to the egg, there's going to be sort of chemicals in the reproductive tract that are going to cause the membrane that covers the head region of the sperm to change. Now, what happens by this is, again, I'm oversimplifying it, but basically this results in the sperm becoming a lot more hyperactive. It realizes, hey, I'm so close to the egg now, so it tries to swim even harder and faster and better to reach its destination, which is, of course, the egg, right? And so finally, by the time the sperm reaches the egg, you really only have maybe a few dozen sperm that are left at this point. And what it needs to do next is to start to try to fertilize that egg, right? It's not one by just meeting the egg, it needs to fertilize the egg as well. So the egg itself has a sort of outer layer or shell, if you will, of cells that protect it. And in order to get past this kind of protective layer of cells, the sperm has to push through to reach that outer layer of the egg known as the zona pellucida. Now, when the sperm reaches the zona pellucida or reaches that shell, they basically attach to these sort of specialized receptors that exist on the surface of the zona pellucida, which will in turn trigger what is known as an acrosomal reaction. Again, we won't get too much in detail of what an acrosomal reaction is, but it's this acrosomal reaction that essentially enables the sperm to really burrow its way in and push its way further in so that it can be on the inside of the zona pellucida. And then ultimately, once inside the zona pellucida, the very first sperm to make contact is going to be able to freeze the egg. And as soon as, fertilize the egg. And as soon as that fertilization has taken place, the outer membrane of the egg and the sperm will fuse. The egg pulls the sperm in into itself, and this essentially kind of modifies the outer membrane a little bit so that other sperm can no longer fuse with it. And essentially that's really just a summary of what fertilization is and how it's occurred. So to really summarize it then, when the sperm and the egg meet, a reaction starts to occur that behaves as sort of, if you imagine kind of like a key trying to open the door. The key is the sperm, the door is the membrane that surrounds the egg, known as the zona pellucida. And eventually the sperm is let in, and then when this happens, the plasma membranes of the sperm and egg are going to fuse with each other. The sperm is going to actually fully enter the egg, and as soon as the sperm is within the egg, the membrane that surrounds the egg is essentially going to get locked up again to prevent any of the other sperm from entering it. So that's it. Right now you have a brand new formed cell that will then go on to give rise to becoming the rest of basically a human being, right? So what we have is now known as a zygote. So ovulation occurred, the egg was released. From the other end, you have the sperm swim up and fertilize that egg to form a zygote. So this image here essentially summarizes every single subsequent step that takes place following fertilization, to the point where you have this ball of cells that's then going to, if you look over here where it says day 8 to 9, you have this ball of cells that's going to be able to implant itself directly into the wall of the uterus so that it can get comfortable for the next 9 months and develop as it should into a fetus, right? So if we go through each one of those steps, right? First off, right off the bat, we described it earlier. Ovulation is basically when you have that single egg be released. That single egg is released from the ovary over here. So again, remember how I said the uterine tube is not connected to the ovary, but then you have sort of chemical release that brings the uterine tube toward the ovary, and you have those fimbriae at the ends of the uterine tube. So the uterine tube comes close enough to the ovary where its fimbriae can sort of pick up the egg that is released. And then you have microscopic cilia lining these walls that are gently going to push the egg down the length of the uterine tube as it's traveling down here, trying to reach, in the event that there is no, in the event that there is a sperm, it's going to get fertilized, and then in the event there isn't, that egg is still just trying to reach the uterus over here. So that's ovulation, and that's the very first step that's happening over here. Next, we have fertilization. Now, as the egg makes its way through the uterine tube, it eventually reaches this sort of ampulla region, A-M-P-U-L-L-A, ampulla region of the uterine tube. And it's roughly at this region over here that it should technically encounter the sperm cell. And this is the part where we discussed earlier, where we said the sperm will meet the egg, they will fuse, and when they do, fertilization occurs, and that new structure that's formed by the fertilization or the fusion of the sperm and the egg is known as the zygote, Z-Y-G-O-T-E. So what's going to continue moving down the uterine tube to make its way toward the uterus is going to be that zygote. So as this zygote, this is what it looks like, and this is sort of the outer shell that protects it all, the zona pellucida that protects it all. Now, as the zygote makes its merry little way down the uterine tube, the cells within it are going to start to proliferate, meaning that the cells within it are going to start to replicate continuously all the time as it's moving down. So what happens next? Next, you have the process of cleavage. So as the zygote is starting to make its way down toward the uterus itself, it kind of goes from being a few cells to many, many, many cells. And this process of proliferation is basically what's known as cleavage, where cleavage, of course, comes from the word cleaving, right? So the cells within the zygote are cleaving or they're dividing. And as this is happening, and you can see in this image here, this zygote has remained the exact same size, but the number of cells within it has started to proliferate. So you go from 2 cells to 4 cells to 8 cells, and so on and so forth. So the cells within it continue doubling in number until you basically get to a point where it is just so densely packed with cells. And because it's starting to look a lot more different, we rename what it looks like now. So then at this stage, you have a compact cluster of cells that resembles what kind of looks like a mulberry. So mulberry is basically, the Latin word for mulberry is basically a morula. And basically that's what this little ball of cells looks like. So the cells within this morula are known as blastomeres. So blastomeres. These are all these individual cells that came about as a result of that cleavage that occurred in the previous step, right? So now you have, it's still the same size, by the way, just packed with cells, and so we call it a morula. Now the cells within the morula, so going from here toward here, you can see again it's starting to look different. Excuse me. So the cells within the morula are going to start to reposition themselves into basically 2 very distinct groups or populations of cells. And because again it started to look a little bit different, we rename it and now we call it a blastocyst. So within the blastocyst, some of these cells are going to gather here in a more sort of central region off to one side, and the remainder of the cells are going to kind of arrange themselves along the periphery. So the cells in the central or inner region are going to be known as the inner cell mass, and these will later go on to give rise to the actual embryo itself, and the cells kind of along the edges or along the periphery of it, this is known as the outer cell mass, and these cells will essentially contribute to the structures that are going to help to support and maintain the growth and health of that embryo. So if we zoom in now, this is the blastocyst that we saw. So if we zoom into the inner cell mass and the outer cell mass. Now the term blastocyst itself, if we look at it in a little bit more detail, blastocyst means, I think it's Greek in origin, and I believe it means a germ or a sprout within a capsule, which is exactly what this structure is at this point. So within that blastocyst, you have the cells that moved around, right? You have the inner cell mass and you have the outer cell mass. Now another term to describe the inner cell mass is the embryoblast. So the embryoblast, or this region here, is eventually what goes on to become the part of the embryo that ultimately gives rise to you and me. And in fact, this embryoblast itself is going to differentiate into 2 very specific layers. So this is known as the bilaminar disc. 2 bilaminar disc. So the bilaminar disc is composed of the epiblast layer of cells and the hypoblast layer of cells. The epiblast is basically this top layer over here, so this upper layer of cells that'll eventually become those germ cells that we named early on in this lecture. So remember, you had ectoderm, endoderm, and mesoderm. Those come about from the epiblast. And this lower layer of cells is known as the hypoblast layer, and this too is just going to contribute to the formation of other supporting structures of the embryo, like the yolk sac, for example, which again, for the purposes of our lecture, we don't really get into detail of the yolk sac and we don't discuss it any further. So that's what's happening with that inner cell mass or that embryoblast. So the cells within this inner cell mass are basically known as being totipotent. So Totipotent, which means that at this point they're basically immature. And that means that as they differentiate, they're going to become more and more and more functional, which means that they will increasingly have the ability to start to specialize into specific types of cells within the human bodies, right? So these cells are going to remain totipotent for a few days, and then ultimately they're going to be able to choose their path, and they're going to specialize into a specific lineage of cells. That's what's happening with the inner cell mass or the embryoblast. Next, we have the outer cell mass or the trophoblast. So we're very quickly getting up to the end of the first week of development, and this little blastocyst is basically looking for a home within the uterus where it can get comfy and spend the next 9 months in. So as the blastocyst eventually kind of encounters the uterine wall, it's going to basically stick to it, right? It's going to adhere to it. So this is our little blastocyst, and this line here kind of represents the wall of the uterine wall it's encountered. So as soon as the blastocyst meets the uterine wall, these cells of the trophoblast, this outer cell mass, they're going to start to implant into it. And again, for the purposes of today's lecture, we don't get into the nitty-gritty of implantation, but just essentially understanding that it's that outer cell mass that helps to kind of implant the blastocyst within the walls of the uterus, and then that the cells that form the trophoblast layer eventually give rise to structures that support the embryo, and this includes, of course, the placenta, which is an organ that allows for nutrient exchange, waste exchange, gaseous exchange between the developing fetus and its mother. And so with that, that's it, right? That's us having come to the end of the first week of development. So if we do a quick recap of what happened in the first week of development. So we had ovulation, right? With ovulation, you had the release of the egg. That's on one side of the female reproductive tract. On the other side of the female reproductive tract, if the sperm are deposited within the vagina, they're going to be able to swim up the female reproductive tract so that they can try to find a way toward the egg, encounter the egg, fertilize the egg, hopefully in the ampulla region of the uterine tube, and then fertilization will occur. And so once fertilization occurs, you no longer just have an egg and a sperm. Instead, now you have what's known as a zygote, right? So what's going to happen with that zygote is, as it's, again, still making its way down the uterine tube, trying to get to the uterus, it's going to start to, or the cells within it, rather, are going to start to cleave or divide in the process of cleavage, where the cells proliferate from 2 cells to 4 cells, to 8 cells, so on and so forth. So it remains the same size, but the number of cells within it get more and more and more and more, so that by the time that same sized structure is now packed with many, many cells, we now call it a morula. And blastulation then occurs, wherein the morula will differentiate into the blastocyst. The blastocyst is basically when, remember, because we said the morula is just densely packed with cells, right? But the cells within the morula start to position and organize themselves. And so the blastocyst forms through blastulation as the morula goes from morula to blastocyst. Now the blastocyst will then undergo even further differentiation, where you have a very clear, distinct group of cells on the outside, known as the outer cell mass, and a distinct group of cells on the inside, known as the inner cell mass. And of course, the outer cell mass, or the trophoblast, is what will help to kind of burrow into the uterine wall, and the inner cell mass, or the embryoblast, is what gives rise to the embryo proper, that will then become the fetus, that will ultimately become you and I. Okay, so let's start off on the second week of development, or day 8 of development. So by the time we get to this stage, the blastocyst has already come into contact with the uterine wall and it has firmly attached to it. Now, at the same time, that inner layer that you had, that embryoblast layer, has also started to differentiate into that bilaminar disc. Remember where you have the epiblast layer and the hypoblast layer. Now, looking at the outer cell mass again, so that trophoblast layer, what happens here is that that outer mass differentiates into its own component parts. And we won't get into the nitty-gritty of all these parts and naming them and talking about what they do, because that is more detail than is required in this syllabus. But basically, each of its component parts have their own responsibilities, and they basically just help to form, super simplified, right? They help to form the blood vessels or the lacunar networks that are going to be necessary for, when that exchange of gas and metabolites is going to happen. During the second week of pregnancy, you also have the placenta forming. And again, we're not going to be discussing the placenta in any sort of detail as part of your syllabus in embryogenesis, but basically it has a vital role in hormone production, in nutrient exchange, metabolite exchange, gas exchange, and it also helps to protect the fetus while it's in utero as well. And then ultimately post-birth, immediately post-birth, the placenta is extruded as well. So that short explanation really concludes our discussion on the second week of development. And if you're keen to find out more, I urge you to email me after this class so that we can discuss it further. But largely for the purposes of this current syllabus that you have, we don't go into any further detail on the second week of development than that. Okay, so if we then go on to the third week of development, we can start to get into the nitty-gritty a little bit of how we go from really just kind of being like a big pile of cells to looking and functioning like human babies. So this next process that we're going to look at is the differentiation of that bilaminar disc that we had, so the two-layer disc, which was the epiblast cells and the hypoblast cells, into a trilaminar disc, so 3 layers, right? So by the time you get to about day 14 of development, or the start of the third week of development, what's going to happen is the process of gastrulation is going to begin. And gastrulation is the main event of the third week of development. And gastrulation is really important because it's what allows that bilaminar disc to become or to differentiate into that trilaminar disc. And of course, this trilaminar disc is important because it's what gives you those 3 primary germ layers. So the ectoderm, the mesoderm, and the endoderm that we've discussed that we've mentioned a few times now. So of course, we spoke about the bilaminar disc earlier as well when we were talking about the inner cell mass. And its 2 layers are here in blue. You have the epiblast layer of cells, and here in yellow, the hypoblast layer of cells. And we'll expand on this a little bit more. Okay, so at this point in development, what's also happening is what is known as cell signaling. So because of cell signaling, with the cells slowly starting to talk to each other, slowly starting to communicate with each other, this little sort of thickening, if you will, starts to form on this blue layer, which we identified earlier as the epiblast layer. And this thickening is basically what's known as the primitive streak. And this is it from 2 different angles, right? Now, this cell signaling on its own is quite a complex process. It's a multi-step process. So again, for the purposes of just embryology and this syllabus and this lecture, we're not going to discuss it. But it's important that you understand that all of these little events that we're talking about that are happening during embryogenesis are essentially occurring as a result of cell signaling, as a result of those cells communicating with each other. So cell signaling, right? And then the primitive streak appears. Now, at the same time, toward the more cranial end of the primitive streak, another structure begins to form, and this is known as the primitive node. So in all fairness, we're not going to discuss the primitive node in any measure of detail just yet, but at this point I kind of wanted to introduce it just so that we know that it's there, that it starts to appear around the same time as the primitive streak, so that when we get to much later on the development of head and neck structures, you're aware of what this primitive node is, or rather when this primitive node appears. So it's really just for completeness sake. So if we discuss it in the future, or if you come across it when you're doing kind of your own revision or reading through embryology texts, you know that it exists, and you know that it's appearing around the same time as a primitive streak, but then we can just place it on the back burner for now. So if we look at that primitive streak itself, right, so this is it in all of these three images over here. Now, the cells at the primitive streak essentially start to invaginate and start to move into the interior portion of the embryo. So you can start to see that what was a full very adherent kind of blue layer earlier in the previous slide, these cells are sort of starting to kind of move in towards that internal portion of the embryo. And what actually is really happening is that this sort of epiblast layer of cells are going to start migrating, they're going to start invaginating, and they're going to start differentiating into what will ultimately give you the endoderm, the mesoderm, and ectoderm germ layers. And again, this is a side note, and it's not necessarily a part of your embryology. In fact, it's not at all a part of your embryology syllabus at all. But for those of you that are wondering how this might happen, how do you go from all these cells kind of being adherent to each other to just kind of being able to move around and to invaginate, right? So the cells near this primitive streak, they're basically going to start communicating. They're going to start secreting chemicals that will in turn trigger a process that's going to kind of stop or inhibit their ability to continue adhering to each other. And because this chemical is telling them that, hey, you no longer need to be able to stick to each other, so they're now able to move apart from each other, or in other words, in more technical terms, epithelial migration is allowed to occur, right? So the cells of the epiblast layer will, because they're no longer adherent to each other, they're going to be able to kind of go through and travel all the way down to where this in yellow we said was the hypoblast layer. So they're able to come all the way through to the hypoblast layer, displace it and replace it, so that the first group of cells that goes all the way through to that hypoblast layer and displaces that hypoblast layer, they'll start to get nice and comfortable in their new location. And so while they were up here, they were called epiblast cells, right? But because they moved down, because they displaced those hypoblast cells, we have to give them a brand new name because they're in a brand new location doing a brand new thing. So now we call them the endoderm germ layer of cells. Now, after the endoderm germ layer has been fully formed, the epiblast cells back at the top, they're buzzing with excitement. More of them want to kind of move or migrate down as well, because they've now managed to create this whole new layer of cells, right? They've managed to create the endoderm germ layer. So as more of them get ambitious and adventurous and they start to try to, want to migrate downwards, the epiblast actually does send more cells down this primitive groove. But when these new cells travel down, they realize that, hey, look, the endoderm germ layer has been fully filled. It's essentially complete. And so what they have to do instead is to set down a sort of brand new layer just on top of the endoderm germ layer. And again, because they're no longer epiblast cells, they've migrated down, they've created this whole new layer. We call this the mesoderm germ layer. And basically what happens to the epiblast layers, the epiblast cells that are kind of remaining at the top. So basically every cell that wanted to leave and travel down the primitive groove was able to do so, right? So the cells that remain at the topmost layer, again, they're no longer epiblast cells, because what made them that epiblast layer, some of them are now endoderm, some of them are mesoderm. So what's left here essentially has to rebrand itself, right? And in doing so, they call themselves the ectoderm germ layer of cells. And this really briefly simplifies the process of gastrulation, which gives rise to what is known as the gastrula. And the gastrula is this new structure that is characterized by this endoderm, this mesoderm, and this ectoderm germ layers, or these three distinct germ layers. And besides gastrulation giving rise to the gastrula, it's also through gastrulation that the axes of the body, so the anterior, posterior, dorsal, ventral orientations of the body are determined. And basically it's this very transformative process of gastrulation wherein that embryo goes from being that super simple, just a bilaminar disc, to now starting to become a much more complex, much more well-organized organism. And that really brings us to the end of the third week of development, with the most important takeaway you should have from this being the process of gastrulation, where first you have the establishment of the three primary germ layers, so the endoderm germ layer, the mesoderm germ layer, and the ectoderm germ layer, as well as the body axes being determined in this third week of development as well. Okay, so now that we have our germ layers, we know how they came about, we know what they are, we've named them, we've seen them, we can start to go a little bit into more detail on each of them and what they give rise to. So we spoke about the ectoderm. The ectoderm germ layer, if we look at the word ecto itself, ectos actually means outside, and of course derm refers to the skin. So ectoderm, of course, would kind of have a correlation to giving rise to structures that are the kind of essentially the outermost layer of the three germ layers in the embryo. And the derivatives of the ectoderm germ layer are going to be both epithelial in nature, as well as neural tissue, and of course your external ectoderm as well. So the ectoderm derivatives are going to be the nervous system, right? Things like the neural plate, which will give rise to the neural tube and the neural crest cell. So it very firmly has a neural tissue aspect to it. And of course, these terms, again, are very new to you. Neural crest cells, neural tube, neural plate. But these are just terms you can start to kind of percolate in your head a little bit, start to familiarize yourself a little bit, because we will come back to them in a lot more detail next year when you do the development of head and neck structures, right? So this ectoderm, it gives rise to neural tissue. It's going to give rise to the spinal cord, the peripheral nerves, the brain. It's also going to give rise to the development of certain sensory organs like the ears, the eyes, as well as the olfactory epithelium. And of course, that's besides its neural aspect, its epithelial aspect is going to give rise to things like the skin, the hair, the nails, as well as some of the glands in the body, and of course the enamel layer of teeth. Now, if you go one layer further in, you have the middle layer, and that is the mesoderm germ layer. And this layer is one of the major contributors to the entire embryo. The mesoderm layer broadly is going to give rise to mesenchyme cells, right? Things like mesothelium, non-epithelial blood cells. But if we look at the mesoderm, and again, this is kind of this bit of information that I'm going to say next is just kind of to prepare you for when we rediscuss the mesoderm in the development of other organ systems. The mesoderm itself can be divided into different regions. So you have the lateral plate mesoderm, the intermediate mesoderm, and the paraxial mesoderm. And each of these different regions of the mesoderm are going to contribute to the formation of the different structures or organs in the body. And it's that process of gastrulation. Remember, during gastrulation, we saw those cells kind of migrating downwards, right? So the time in which, or the point in which the epiblast cells entered and invaginated downwards, as well as the direction of their subsequent migration, that's what determines the ultimate fate of these mesodermal cells as to whether they'll be lateral plate mesoderm or paraxial mesoderm or even intermediate mesoderm. And so So the paraxial mesoderm, for example, if we look at right over here in red, this is going to get organized into segments known as the somites, which will give rise to the sclerotomes and the dermatomes. And this we will kind of come back into when we talk about the development of the musculoskeletal system. So things that give rise to, the development of the vertebrae, the development of the intervertebral discs, the ribs, the connective tissues, the myotomes here give rise to or develop into myoblasts that will contribute to the formation of the skeletal muscles in the body wall, the muscles in the back, or specifically the intrinsic muscles of the back, your intercostal muscles into the ribs, in between the ribs, and all of this we kind of will keep looking at as we come to discuss the different development of the different organ systems in the body. So that's a paraxial mesoderm, a lot of derivatives. The intermediate mesoderm is going to give rise or differentiate into urogenital structures, so things like the kidneys, the gonads, and their associated structures. Whilst the lateral plate mesoderm is going to differentiate to form sort of the sides of the body, the lateral body wall folds, as well as the dermis of the skin, the limbs, the bones, the connective tissue of the limbs specifically. And so there are a lot of derivatives to this mesodermal germ layer, and I'll leave it to you to kind of read up a little bit more on it to kind of find out the full extent of the reach that it has in terms of the derivatives that it gives rise to. And of course, as I shared earlier, as we do each of the various organ systems, each time a system block starts, I'll come in the beginning and set down the sort of tell you how that organ system develops and what are the sort of different germ layers that give rise to the various structures associated with that organ system. Okay, so mesoderm, major contributor to bones, to cartilage, to connective tissue of the skeleton, to the skeletal muscles, the smooth muscles, a lot of the cardiovascular system, the lymphatic system, the urinary and reproductive systems, as well as linings of the body cavities. So mesoderm really is quite a large contributor to the various structures in the body. Now, the last layer, the last germ layer that we have is the endoderm germ layer, right? So again, we had a trilaminar disc with 3 layers. So we spoke about ectoderm, we spoke about mesoderm, and now we're left with the endoderm germ layer. Now, the epithelia that's derived from the endoderm germ layer is going to eventually line the sort of internal regions of the body that come into contact with external substances. So if you think about it a little bit, external substances like the food that we eat, right? So the internal parts of the body that come into contact with external substances, meaning the gastrointestinal tract, the lungs, because when we breathe, right, the respiratory tracts. So to kind of start to visualize it, and again, this obviously makes more sense as you kind of progress along with your study of embryology. As all of this is happening, you also have embryonic folding that's happening, and so the endoderm ultimately ends up kind of moving towards a sort of midline region and fusing together to give rise to that primitive gut tube. Because again, you have to kind of, if you start to visualize it, where you were just a flat pancake of cells, this embryo is now becoming a little bit more three-dimensional. Its sides have to come together through embryonic folding, and that's what ultimately gives rise to the primitive gut tube. So the derivatives of the endoderm germ layer include the epithelial lining of the respiratory tract, things like the liver, the pancreas, the urinary bladder, the urethra, some of the glands, like the thyroid, the parathyroid glands, as well as structures of the developing digestive system. So the foregut, the midgut, and hindgut structures, which again are things that you will keep sort of reinforcing as you do the other system blocks. Okay, so that is really the germ layer the three germ layers and their derivatives. Next, we'll have a look at embryonic folding and talk about how through the process of embryonic folding, we go from being this two-dimensional flat sheet of developing cells to this sort of group of cells that's three-dimensional and starting to slowly, day by day, look a lot more like a fetus. All right, okay, so let's go back to our trilaminar disc for a second. So trilaminar, of course, referring to the endoderm, the ectoderm, and the mesoderm germ layers. And disc, of course, kind of implies to us that those three germ layers are very much arranged in a flat, sort of oval type of sheet, or like in a disc, right? So as all of this differentiation is happening within this developing embryo, and as all of this growth is happening with this developing embryo, what's going to start to happen at the same time is what used to be a flat embryo is going to start to fold in different planes or in different ways, so that it starts to get a little bit more looking three-dimensional. So it's going to specifically start folding in a longitudinal direction as well as a lateral direction. Now, if we talk about what is known as the cephalocaudal, cephalo, of course, referring to the cephalic region or the cranial region, and caudal referring to the sort of more inferior or the more leg region, so cephalocaudal or head-tail region folding, that's going to bring this flat embryo, the sides of it together in that head-tail direction. It's going to bring that head and tail or the developing legs closer to each other so that as a result, you're starting to get that sort of curved appearance of the embryo in utero that you're familiar with by the time it gets to that fetal stage of development. So that's what's happening with your longitudinal folding. Now, if you think about what's happening with the lateral folding, so the sides of the embryo, right? So that's going to bend the embryo in a sort of right-left axis to start to create a more sort of cylindrical shape or cylindrical body shape for the embryo. So the sides of the embryo are going to come closer together. They're going to fold to where the left side edge and the right side edge are able to sort of meet in a midline region and bring all those three germ layers, because again, if you remember, we're talking about a flat disc. So if they start to come together in a midline region, it also brings those three germ layers together to allow for that continuity. And this continuity that it creates is kind of what's represented in that bottom third image in blue and red and yellow. It's how you're getting to see that idea of a tube forming within a tube, right? So we're sort of growing as this smaller cylindrical structure within a larger cylindrical structure. And that inner tube would be, of course, the developing gut at this point, with the outer tube, of course, just kind of representing the whole rest of the body, so like the body walls and everything else. And so that this kind of ensures that by the time we actually get to the end of embryonic folding, so we're no longer just a flat disc of cells, but rather we're getting to have this more three-dimensional, more cylindrical appearance. We've also created a couple of things. So first off, we've created the primitive gut tube, and the primitive gut tube will later on, of course, as development kind of progresses, that primitive gut tube will become the actual more mature gastrointestinal system. But we've also created the amniotic cavity at this point, which is going to very comfortably envelope the embryo to provide it with protection through its development through the fetal stage and through the rest of its time in utero. And we've also very much given rise to the yolk sac, which is going to provide, in these early stages at least, it's going to provide nutrition, it's going to provide blood cell formation to kind of nourish this developing embryo in these early stages before that role is going to be taken over by the placenta instead. And that quite simply really just very summarized describes what embryonic folding is like and how we go to being from, a flat sheet of cells to very much being that sort of three-dimensional cylindrical shaped structure. which means that at this point, we're at the end of the 8th week of development, right? So following the embryonic period of development, we have the fetal period of development. So running from the start of week 9 all the way through to the point of birth, right? So during the fetal period of development, you have a lot of large changes that occur in terms of rapid growth in size of the embryo, of the fetus, rapid growth in mass and complexity and in proportions of this fetus. But you don't so much have large changes happening in the form of the embryo itself. Rather, all the organ systems are developing, as you'll see, when we do other parts of this lecture series. But rather, by the time you get to the fetal stage of development, or the start of the 9th week of development, this fetus is just needing to get larger, more mature. So what you notice during this period is basically an increase in size, especially of the inner organs, which of course leads to then the overall growth of the fetus itself, as it starts to kind of stretch, itself out. As you can see in this transition here, it starts to stretch itself out and take on that typical, more sort of baby-like shape that we're familiar with. So again, this fetal period of development starts at the start of the 9th week of development, all the way through to birth of the fetus, which occurs between 37 and 42 weeks after fertilization. So if we talk about weeks 9 to 15, here you have the development of the reproductive organs, the eyelids forming, tooth buds start to appear, and a little bit of brain activity can be detected at this stage as well. By the time you get between weeks 16 to week 26 of development, you have a lot more rapid development of the brain. The muscles are developing, the eyebrows are coming in, the lashes are coming in, the nails are appearing, and then in the lungs, the alveoli are starting to develop. And this you'll see when we talk about the embryology of the respiratory system. Between these weeks 16 to 26, you also have the internal parts of the eyes and the ears developing further as well. So that when you finally get to week 27 to week 38 of development, this developing fetus is going to have an incredible increase in body fat and therefore obviously weight. And also the major organ systems will start to become functional, bone development will complete, really just preparing this fetus to be able to sustain its life outside of the uterus. And so basically at the stage, to complement the rest of the gross anatomy studying that you're going to be discussing during this phase 1 of your program, I thought I'd briefly discuss the process of ossification as well. And so ossification in and of itself is basically the process through which your bones start to form. And this kind of starts to tie in with what you're learning in your histology lectures and practicals, as well as, of course, through your study of gross anatomy. So at this stage, this sort of discussion of ossification is a more sort of brief introduction to ossification, because of course, like I shared at the start of this lecture, we do come back to ossification and we end up discussing it in significantly more detail when we talk about the embryology of the musculoskeletal system in the first semester of your phase 2 syllabus. Okay, so if you remember, when we spoke about the trilaminar disc and its very many layers, or rather its 3 layers, endoderm, ectoderm, and mesoderm, we said that the mesodermal cells are what are responsible for the formation of bones and cartilage in the body, correct? So if we talk about bones specifically, or ossification, it can occur in one of 2 ways. So we can either have intramembranous ossification, or we can have endochondral ossification. Now these drop-down sort of lists are a good summary of these 2 processes. So essentially, if we're talking about intramembranous ossification, basically we're talking about mesoderm cells giving rise to bones, and that's it. That's literally it. That means that wherever in the body there are mesodermal cells or mesenchymal cells that are fated to be bones in that region, those mesenchymal cells will essentially differentiate and produce bones. So they go from mesenchymal cells to osteoblasts to osteocytes. That's it. That's intramembranous ossification. But if we talk about endochondral ossification, here the mesoderm first has to kind of develop into cartilage, and then that cartilage will eventually disintegrate. And in disintegrating, it leaves an empty space, and into that empty space will come in those bone-forming cells to fill the area in and to replace what used to be cartilage with bone. And what's important about endochondral ossification is really is the fact that it's how a lot of our long bones, especially in the upper limbs, the lower limbs, how they develop is through endochondral ossification, with the exception, of course, the clavicle, so your shoulder bone, your collarbone, sorry, over here, which is technically a long bone, but it is formed through intramembranous ossification. So this is just a super, very watered down, in a way, brief summary to give you a kind of, to explain the sort of major difference that exists between intramembranous ossification and endochondral ossification. All right, that's it. With that, we've come to the end of our lecture. So to give you a sort of summary of what we've discussed today as part of the different stages of embryogenesis, right? We started off by talking about fertilization. When we spoke about fertilization, we also spoke about how cleavage happens, how morula formation and blastocyst formation happens. So again, when the sperm first meets the egg, it fertilizes it, and as that fertilized zygote starts to make its way down the uterine tubes, the cells within it will cleave, up until which point where, whilst it hasn't grown any larger in size, it's going to be, the cells within it have proliferated so much that it's compacted to where it looks like a morula. The cells within that morula, or the blastomeres, reposition themselves to give rise to the blastocyst. And of course, we spoke about the formation of the trilaminar disc, which occurs through the process of gastrulation, wherein that blastula becomes a gastrula, so that bilaminar disc, so that epiblast layer and the hypoblast layer goes on to give you a trilaminar disc with the with the three layers being the ectoderm germ layer, the mesoderm germ layer, and the endoderm germ layer. Next, we also discussed embryonic folding, and this occurs either in the longitudinal plane or in the lateral plane, where we go from being that flat pancake of cells, or that flat disc of cells, to becoming a more sort of cylindrical shape, a more sort of three-dimensional shape. And we finally wrapped up by talking very briefly about ossification and how intramembranous ossification describes direct bone formation, whilst endochondral ossification describes bone formation first from a cartilaginous model. So at this point, I'd like to introduce you to a very handy reference tool that I've personally created myself for you. I highly recommend you get on learnanatomy.com and use it as a guide or a resource to your study of anatomy. It's all encompassing, so it's going to include all of the anatomy learning you're going to be doing within the next 2 years. Besides the gross anatomy aspects, it also includes all of the embryology lectures that we're going to be doing in the next 2 years. And it's handy because it's really a good supplement to all of the gross anatomy lectures, as well as the embryology lectures that you're going to be attending. And it really helps to kind of tie in that information for you together. There's also a little tab on the website where you can ask me questions anonymously, and we can have a little bit of a discussion, be it about embryology or about gross anatomy. And it's a really useful tool that I recommend you use. Okay, so thank you very much for your time today and for attending our very first embryology lecture on embryogenesis and this How to Build a Human lecture series. If you have any questions about today's lecture, I hope you'll feel free to contact me directly either at antskr@nus.edu.sg, or if you prefer, you can click that little bit.ly link that says Ask Questions Anonymously and drop me a little question anonymously, and I will compile these questions and kind of send them out to the rest of your class so that everyone can make use of them. Again, thank you for your time. I hope you have a good rest of your day, and I'll see you soon. Bye, everyone.